All right, well, it's a great pleasure and honor to be here today to tell you about some of our latest work in developing novel approaches to producing animal protein in a more sustainable way that has sustainable potential. All right, so the vision that really drives our work is to address some of these major societal challenges, which we've heard much about in the last couple of days. This very intertwined challenges of advancing human and planetary health along with social justice. And in my own work, I'm doing that. So the major research question that drives our work is to really understand how cells respond to physical mechanical cues in their environment to generate an integrated response. And if we could fully understand the molecules and pathways that are involved in regulating cellular response to these mechanical cues, it would really enable us to control and also predict cell behaviors for a spectrum of applications spanning human and planetary health. And in my own lab, we're addressing this question by building new technologies and platforms to be able to measure in higher throughput cell mechanical behaviors. We're working to understand the fundamental mechanisms that regulate these behaviors, and also translating these findings for applications in both cancer and also in sustainable food. And that's what I'm here to tell you about today. So the vision for that really drives this arm of our work, which we've been working on for the past few years, so it's a newer direction in our research program, is really to develop nutrient-rich food that can be available and accessible for everyone. Meat is a very nutritious food for many people around the world, but in the face of extreme climate events or supply chain disruptions that many of us have experienced over the past few years, we're at risk for having nutrient-rich foods become very expensive and a luxury item. So having sort of multiple sort of ways of producing animal protein could help to build resilience in our food supply chains. There's also increasing demand from consumers these days for foods that are both nutritious but also support planetary health and have reduced environmental impact. And as you can see from many of you've experienced the emergence of plant-based meats, which are very popular, but the majority of consumers still want to eat real meat. So this is where cultured meat, the process of using cells extracted from a living animal and growing them in culture to form meat products, can be a potential solution. And there's still lots of work ongoing to examine the environmental implications and also technical feasibility. But our data so far is suggesting that cultured meat can be a potential solution to creating a sustainable source of animal protein. It's been mentioned also the IPCC report, which also sort of highlights the importance of novel foods to address some of these climate goals as well. Cultured meat may strike you as a new idea, but it's been around for quite some time, originally described by the concept by Churchill, which really kind of got at the question of why we couldn't grow just individual pieces of meat without the whole animal, which can be more energetically wasteful. But fast forward nearly 100 years, and you may have heard this news last week that in the United States, the USDA has now just approved the sale, making it the second country after Singapore to be selling cultured meat products. This may give you the impression that we've solved all the problems. We haven't. This meat is only available at a very small scale, and so there's still major challenges to be solved in research and development. So to give you a sense, typically we culture cells in a very small petri dish. The total mass of cells in a petri dish would be on the scale of milligrams, but to produce cells for food, we'd need orders of magnitude higher mass of cells. And this can be achieved by just using a larger number of Petri dishes, which is what was done by Mark Pust, who pioneered this field back in 2013 for the first proof of concept cultured meat burger. Of course, this is not sustainable. So growing cells in larger bioreactor vessels is a very attractive solution. So there's various challenges in culturing meat on such a large scale. People in labs around the world are working on addressing the media, the source of cells, and also how do we generate foods that people would want to eat, that are delicious and that taste delicious and have a great texture. And with my background in mechanobiology, this is exactly the question that we're seeking to address. So we know from foundational work in mechanobiology that these mechanical cues that regulate cell behaviors are really important for driving some of the phenotypes important for cultured meat. You can see from this work from Penny Gilbert that cells, muscle cells taken from mouse grow much more extensively on a softer 12 kilopascal hydrogel rather than on the stiffer Petri dish, which is shown on the bottom. We also know from work of Adam Engler and Dennis Disher that myoblasts cultured on a sort of medium stiffness substrate of around 11 kilopascal are more likely to fuse and form myotubes, which are important for the development of skeletal muscle. So you can see from the alignment of these blue nuclei. So taking these findings, we've been developing customized scaffolds to grow both muscle and also fat tissue for cultured meat applications. So to give you an overview, our approach is to make tiny scaffolds that can be also compatible with culture in a suspension bioreactor. And the idea is to harvest these tissues into a meat product. So we use emulsions, just the way you would make a salad dressing, water and oil, to form a template for these tiny microbead particles that look like this. We can control the stiffness just by the amount of polymer that's in these beads. For proof of concept, we've used gelatin. We can also replicate this grooved structure like natural skeletal muscle by making this template that we sort of squish the beads between. This makes these grooved microcarriers, which we can also modulate the stiffness of as well. So the question is, can the cells grow on these? And we use mouse CTC12 cells as proof of concept here. You can see that the cells, the gelatin is shown in yellow. The cells have cyan-colored nuclei, and we compare these to commercially available cytodex beads. Over time, we see that the number of cells tends to increase over time, suggesting that cells are growing very nicely on these edible microcarriers. And we've quantified the growth of cells by quantifying genomic DNA as well, which you can see that the cells on the edible microcarriers shown in pink and blue are increasing in number even more compared to the commercially available microcarriers. So can these cells also differentiate to form the beautiful myotubes that are important for muscle structure? And shown here in the magenta is a myosin heavy chain stain. So you can see these myotubes actually can span hundreds of microns through these aggregates of cells and these microcarrier particles. It's not quite like the skeletal muscle shown here by the rat, but they're much longer than the cytodex commercially available microcarriers. What was interesting is that we found these aggregates of these microcarriers and the cells forming in suspension culture, and you can see across the different edible microcarriers, these structures forming. Again, this is with mouse cells, so who really wants to eat mouse cells? There is a market, no joke, for pet food, cultured pet food. But we also tested the use of bovine satellite muscle cells and showed similar effects. We're able to harvest these microtissues into a larger cohesive structure that can be cooked, shows signs of browning characteristic of myoid reactions. So what about the fat? We know fat is really important for the flavor, the mouthfeel. This is just showing the sort of characterization of Wagyu beef with increasing marbling being more desired. You can find this in cultures around the world. So we also were able to culture cells, both primary rabbit adipocytes and mouse 3T3L1 adipocytes on these beads. The beads are shown in cyan and the nuclei in blue. And the lipids you can see accumulating in yellow for both the rabbit and the mouse. We were then able to integrate these fat microtissues into layered structures with our muscle microtissues. And you can see here a cross-section of cultured rabbit meat, where the blue is showing you the protein component, the muscle component, and the cyan are those microcarriers from the fat component. Shown on the right is false color, just for easier visualization, the fat in yellow and the red is the muscle. Compared to Wagyu beefsteak, you can see a similar layered structure, albeit the length scale of the muscle component is different. But importantly, this is proof of concept work for our cultured meat that shows the feasibility of creating these structures. And what's really interesting is that we found these multi-component structures happen by spontaneous adhesion between these microtissues, which results in a cohesive, mechanically stable structure. And we've begun to mechanically characterize these cultured meats, comparing them to Wagyu beefsteak, and we are seeing similar on the order of magnitude mechanical properties, for example, ultimate tensile strength. The scale is still small. You can see by the size of the sesame seeds on the bun, but we're working to scale up, and that's really, I think, the realm of more commercial opportunities. So today, I've shown you that we can grow muscle and fat microtissues in suspension culture using these edible microcarriers. I think this has potential as a scalable process for culturing cells for food applications. We're able to generate these multi-component tissues and pattern these tissues just by spontaneous adhesion. And I've shown you we can translate the approach to different types of cells as well. Beef, rabbit, and mouse. I didn't show you some of the work from our pectin microcarriers, but we see similar behaviors on plant-based microcarriers as well. So I think another exciting opportunity here is to highlight some of the more fundamental research questions that come out of some of this work. For example, how do these principles of mechanobiology translate across species? A lot of the optimal stiffness that's been discovered for accelerating cell proliferation and differentiation, for example, it's been largely conducted on mouse models. And so I think here there's opportunity to understand how broadly do these principles of mechanobiology apply across different species. Furthermore, I think it's really will be essential into the future to understand how cells integrate different signals from different mechanical stimuli, for example, the matrix stiffness, together with the fluid shear stresses that cells might experience in suspension culture. So this is towards our vision to be able to create cultured meat, both ground meat and mixed, but also structured cuts of meat. And I'd just like to highlight some of our ongoing work that I think really uses and translates some approaches from the life sciences to address these questions. Specifically, we've been working in long-time collaboration with Robert Damoiseau of the Molecular Shared Screening Resource at UCLA. So we've been now screening libraries of small molecules and potential food ingredients to better understand how we could what sort of soluble additives we could include in the media, and also to understand more fundamentally the mechanisms and pathways that might be involved in regulating, for example, lipid accumulation in adipose tissues. Of course, there are many outstanding challenges remaining. This one's come up already, is public perception and consumer adoption. And so to address this question, we've been working with our behavioral scientist collaborator, Tomiyama at UCLA. And what's interesting, I think, to us is that there's lots of challenges one can envision for these novel food products, but also challenges that we can address through fundamental research in the life sciences and also further research and development, such as understanding how we can make a better tasting or a better textured piece of cultured meat. Last but not least, I think engaging people in critical conversations about novel foods to encourage this transparent dialogue will be really critical. And to this end, I've been hosting these events over the past 10, 12 years now to engage public in these conversations. Here's one on people, food, and climate that we hosted last year that brought together architect, a chef, and heirloom corn entrepreneur and regenerative agriculture scientist and soil scientist. So you can find these available on YouTube, Science and Food on YouTube. So just to quickly finish, acknowledging some of the people, Stephanie Kveski here, graduate student, has really been a major proponent and spearheading this direction of research in my group. And of course, funding sources specifically for the cultured meat, the USDA, the NSF, and also the philanthropic sources such as the Noble Family Foundation Fund at UCLA. Thank you very much.